Welcome to BioTales. So today, we're starting with someone you might not immediately connect with Biotech, a filmmaker.
Right. Someone behind powerful documentaries. You might have watched the documentary film The Price of Sugar.
Exactly. That exposed the really harsh conditions in the Dominican Republic sugar trade, and also Jim Allison, Breakthrough.
Ah, yes, The Immunologist. That film got a lot of attention, didn't it? Washington Post top 10 of 2019, I think?
That's the one, a real scientific maverick.
Yeah.
But here's the twist. That same filmmaker, the producer behind these documentaries, is also the co-founder and CEO of a biotech company.
Okay, that's definitely not your typical career path.
Not at all. And this isn't just a side project. There was major news just recently, August 18th this year. The German giant Merck KGAA announced a huge partnership, potentially worth over $2 billion.
Wow. With this filmmaker's company.
Precisely. The company is Skyhawk Therapeutics, and they're focused on creating new drugs for some truly devastating neurological disorders.
So a filmmaker, a $2 billion pharma deal, and neuroscience. There's definitely a story there.
That's exactly what we're exploring on BioTales today. We're unraveling this really compelling story of how a filmmaker, a brilliant scientist, and a seasoned drug developer came together
Their goal being?
to essentially industrialize the editing of our genetic code. This is the origin story, the journey of Skyhawk.
Okay, I'm intrigued. Let's dive in. How did this vision even start?
Well, let's go back to the early 2000s. Picture Bill Haney. One moment he's, knee-deep in an editing bay, crafting these intense documentary narratives.
Right, immersed in the story.
Then, maybe hours later, he's in a completely different world. A boardroom, pitching a tech startup.
So juggling two very different lives.
It seems like it, but Haney saw a connection. He argues it's all about one core skill. Producing.
Producing, like
Yeah.
movie producing.
Exactly. Think about what a great producer does. They find a powerful story, right? They assemble the absolute best cast and crew, secure the funding.
And clear roadblocks, get things done.
Precisely. And Haney believes that craft, that process, is fundamentally the same, whether you're making an Oscar contender or developing a first-in-class therapeutic.
That's a really interesting perspective, applying a filmmaker's producing mindset to biotech.
And it's absolutely key to understanding Skyhawk's whole strategy.
Yeah.
Now, let's talk about the SMARC, the initial idea.
Okay. Where did that come from?
Haney was on a humanitarian trip back in the Dominican Republic, the world he'd shown in The Price of Sugar.
Ah, so connecting back to his film work.
Yes. And it's there he meets Kathleen McCarthy, a
Yeah.
scientist. And she has this idea that's, well, radical.
Okay, what was this radical idea?
Kathleen's focus was on something called RNA splicing. And her profound insight was this. Many devastating, untreatable diseases might not stem from a faulty gene itself.
But from a faulty reading of that gene?
Precisely. Errors in the splicing, like a rushed editor, accidentally cutting out crucial sentences, or maybe leaving in garbled instructions.
And these aren't just minor typos, presumably. What kind of impact are we talking about?
We're talking devastating impacts, especially in neurological diseases. Think about frontotemporal dementia.
Okay.
In some forms, a specific missplicing of the tau gene leads to the production of these really harmful proteins that damage brain cells.
I see. A direct link from splicing error to pathology.
Or another example, spinal muscular atrophy.
Terrible disease affecting motor neurons.
Right. And in many cases, it comes down to the splicing machinery skipping just a single critical segment, an exon, in the SMN gene transcript. Without that exon, you don't get enough functional protein, and motor neurons die off.
So McCarthy saw this pattern, these splicing errors, as a common thread in neurodegeneration, maybe even other areas like cancer.
Exactly. She was convinced that these cellular editing mistakes were the root cause, or at least a major driver, of many really difficult illnesses.
Okay, so she identified this critical problem space, but the big question then is, how do you fix it? Especially with a small molecule drug, which was her idea, right?
Yes, that was the elegant part. While a lot of the field was looking at complex biologics or even gene therapies, Kathleen believed the answer could lie in small molecules, orally available pills that could potentially cross the blood-brain barrier.
Okay, but how? How could a small molecule achieve that kind of precision, influencing which bits of RNA get spliced together? That sounds incredibly difficult. RNA is notoriously flexible.
It is difficult, and that was the skepticism they faced. But the idea wasn't brute force. These molecules aren't meant to, rip apart the RNA. They act more like molecular chaperone.
Chaperones guiding the process.
Kind of. They're designed to bind very specifically to certain structures, unique folds or loops on the pre-mRNA molecule near a splice site.
Okay, so they find a specific address on the RNA strand.
Exactly. And that binding event acts like a subtle signal. It can attract the good splicing factors, the proteins that tell the cellular machinery, hey, splice us some, you need to include this exon here.
Waving a flag.
Right. Or conversely, the small molecule might bind in a way that physically blocks negative factors that would normally tell the machinery to skip that crucial exon.
So it's nudging the cell's own system.
Precisely. These are modulators, whispering instructions to the cell's own highly sophisticated editing suite, just guiding it to make the correct, healthy version of the protein.
That's a subtle but powerful mechanism, using small molecules to achieve that kind of RNA modulation.
Yeah, that was definitely pushing the boundaries.
As Kathleen laid all this out, Bill Haney, the producer, he immediately saw it. He saw the movie.
The movie? Yeah. Missplicing is the villain. These small molecules as the potential heroes. He saw the narrative arc.
Act 1, discover the target. Act 2, design the molecule. Act 3, prove it works in the clinic.
Exactly. And the potential for a blockbuster ending, a truly life-changing medicine. He got the story.
But a great story, a great vision, needs a team to actually make it happen. They needed the scientific expertise to execute.
They did. They needed someone who really knew the chemistry, the biology, the pitfalls. Someone who had, in Haney's terms, made movies before in this specific challenging area.
And who was that?
That's where Sergei Pashkin entered the picture. He was a veteran, someone who'd been involved in the very first attempts years earlier to try and drug RNA splicing.
So he knew the landscape, knew what hadn't worked.
He brought his scar tissue. Invaluable, hard-won wisdom from past failures. Knowing what not to do is sometimes just as important, right?
Absolutely critical in drug development, avoids repeating costly mistakes.
So with Sergei on board, the founding trio was complete. You had Kathleen, the visionary scientist with the core idea, Bill, the producer, driving the strategy and narrative, and Sergei, the seasoned expert who knew how to navigate the complex science.
A strong foundation, vision, execution, experience.
Okay, so they have the team, they have the vision. Next big hurdle? Money, capital.
And pitching this in 2018, drugging RNA with small molecules. That must have been a tough sell.
It really was. As you said, RNA was still considered fringe by many VCs. Too dynamic, too floppy, no clear pockets for drugs to bind stably. That was the dogma.
So how did Haney get traction?
He did what producers do best. He didn't just pitch the science, he told a compelling story. He focused on the immense unmet human need, the potential impact on patients' lives.
Appealing to the heart as well as the head?
And it clearly resonated. They put together a pretty unusual syndicate for their first seed around about $8 million.
Unusual how?
Well, it included some traditional biotech VCs, sure, but also people like Tim Disney, that's Walt Disney's great nephew, and even the Duke of Bedford.
Okay, that is eclectic. Shows the power of a good narrative, I suppose.
A filmmaker knows his audience, right? And that initial backing set the stage. Later, they raised a massive $100 million equity round and secured a $60 million upfront payment in a deal with Celgene.
So they built a serious war chest fairly quickly. What did they build with it? What is their platform?
Their platform is called SkyStar, essentially their engine, a really powerful kind of closed-loop system for discovering these RNA-modulating small molecules. It has two main parts that constantly feed information back and forth. First, there's a massive computational engine.
Okay, using computers, AI, machine learning.
Exactly. It crunches through just enormous amounts of genomic data, structural data, looking for those specific RNA shapes, the hairpins, the loops, across the entire transcriptome that might be druggable. It predicts potential targets.
A target finding machine, basically, hunting for those specific addresses on the RNA.
You got it. Then, the second part is the high-throughput wet lab.
Where the biology happens.
Right. This is where they take the predictions from the computer and test them at scale. They need to confirm, are these predicted sites actually modulatable? Can we really alter the RNA's function with this small molecule in living cells?
So, bridging the gap between prediction and biological reality. Very important.
And here's the clever part. Every single experiment, whether it works brilliantly or fails completely, the data gets fed right back into the computational models.
Ah, I see. So the successes and the failures make the prediction engine smarter over time.
Exactly. The experiments train the models, and the smarter models guide better experiments. It's synergistic. The analogy they use is it's not just about finding the needle in the haystack.
It's about making the haystack smaller and smaller with every try.
Precisely. It refines the search space continuously.
Okay, that makes sense. And the goal of these drugs, sometimes it's fixing a splicing mistake, right? Making sure a needed exon is included.
Yes, that's one major goal. Force the inclusion of a missing piece to make a functional protein. But there's another strategy they use. Sometimes the goal is to intentionally introduce an error.
Introduce an error. Why would you want to do that?
Well, imagine you have a gene producing a toxic protein. Instead of trying to fix the protein, you go after the faulty message before it gets translated.
Okay.
Skyhawk can design molecules that force the splicing machinery to include a little extra piece of code, one that contains a premature stop signal.
Like putting a period in the middle of the sentence.
Exactly. And the cell has its own quality control system. It's called nonsense-mediated decay. This system recognizes messages with these premature stop signals as flawed.
And it destroys them.
It flags them for destruction. So the faulty RNA message gets eliminated before it can even be used to make that toxic protein.
So it's not just fixing the message, it's deleting the bad message entirely. That's elegant.
Very elegant. A sophisticated way to silence a problematic gene's output.
Okay, so they have this powerful platform. What was its first big test? Where did they apply it first?
The first major test case, the first real disease target they went after publicly, was Huntington's disease.
A devastating neurodegenerative condition. Makes sense given their focus.
Right. And to understand their approach, maybe let's quickly recap the basics of Huntington's.
Sure. It all centers on the HTT gene, which normally makes a protein called Huntington, important for brain cells.
But in people with Huntington's, that gene has a sort of genetic stutter. A small segment of DNA code is repeated way too many times. And this leads to the production of a misshapen, malfunctioning version of the Huntington protein. This toxic mutant Huntington clumps up and damages neurons over time.
And there's another insidious aspect too, isn't there? The instability.
Yes, that's the second problem. That stutter, a CAG repeat, isn't stable. In certain cells, especially brain cells, it can actually get longer over a person's lifetime.
Worsening the disease progression, a double whammy. Toxic protein and increasing genetic instability.
Precisely. It's a really tough biological problem.
So how did Skyhawk's drug, Sky0515, aim to tackle this? What was the strategy?
Well, Sky0515 came out of the SkyStar platform, and it was designed with a really insightful dual-action approach.
Dual action? How so?
So first, it uses that clever nonsense-mediated decay strategy we just talked about. It targets the pre-mRNA of the mutant HTT gene.
Okay.
It binds and forces the inclusion of a segment containing one of those premature stop signals.
Flagging the toxic message for destruction. Got it.
But it doesn't stop there. The molecule was also designed to reduce the production of another protein called PMS1.
And why target PMS1?
Because PMS1 is known to be a key driver of that genetic instability, the expansion of the CAG repeat.
Ah, so it's a two-pronged attack. Reduce the existing toxic protein and potentially slow down the underlying genetic problem that makes the disease worse over time.
That's the idea. Tackle both the symptom and a driver of progression.
That's quite ambitious for a single small molecule. And what about getting it into the brain? That's always the challenge in neurology.
This is where the small molecule approach really shines, potentially.
Yeah.
Sky0515 is an oral pill. And the SkyStar platform is designed from the ground up to build in the properties needed for brain penetration. Things like low molecular weight, the right level of greasiness, chemists call it lipophilicity.
Making sure it can slip through the blood-brain barrier.
And also designing it to avoid the brain's natural defense mechanisms, these efflux pumps that try to kick foreign molecules out. It's all part of the design criteria.
A huge potential advantage over larger biologic drugs for brain diseases. Did they see early signs it was working?
They did. Early phase 1 trials were promising. They showed not just safety, but also evidence of target engagement in humans, with reports of up to about a 72% reduction in the levels of the target HTT mRNA.
A significant reduction. Okay, so the platform was delivering, the lead drug looked promising. What happened next?
Well, Big Pharma started paying very close attention. What followed was this, almost a torrent of partnership deals.
We mentioned Merck KGAA recently, but who else came knocking?
Let's see. Celgene was early, back in 2018. Then Biogen and Takeda in 2019. Merck, that's Merck & Co., the U.S. one, also in 2019. And they expanded that in 2020. Vertex came in late 2020, Sanofi in 2022, Ipsen in 2024. And you have to see this sequence of deals culminating in that huge Merck KGAA pact as the ultimate external validation for their platform and approach.
No kidding. So boiling it down, why was Skyhawk so irresistible to these giants? What was the core strategic appeal?
I think there are a few key reasons. First, it's about gaining access to a totally new frontier.
Meaning?
Meaning big pharma pipelines are often full of traditional drugs, small molecules hitting proteins, antibodies. But many diseases, especially neurology, have proven undruggable with those tools.
Right, the targets are just inaccessible or the biology's too complex.
Exactly. Skyhawk's RNA splicing modulation. It's a fundamentally new modality, a new way to drug targets. As Amy Cao, Merck KGAA's head of neuroscience research, put it, it aligns perfectly with their focus on innovative science and next-generation technologies. These deals are like buying a ticket to the future of drug discovery.
Okay, access to innovation. What else?
Second, it's about de-risked innovation. Building a platform like SkyStar from scratch. That's incredibly expensive, takes years, and is super risky. Will it even work?
A huge gamble for a large organization.
Right. So it's often more efficient for big pharma to let nimble biotechs like Skyhawk do that initial high-risk pioneering work. Skyhawk took the risk, built the engine, and showed it could produce drug candidates.
And pharma pays a premium to access that validated, de-risked platform, fills gaps in their own R&D pipeline.
Exactly. Especially, as Ipsen mentioned when they did their deal, for diseases with no approved therapies. It's a way to quickly enter high-need areas.
Makes sense. And the third reason?
I think it comes back to the type of drug Skyhawk produces. We talked about it with Sky0515. They make oral small molecules designed to cross the blood-brain barrier.
The holy grail for neuro drug development.
Pretty much. In a world increasingly filled with complex, injectable advanced therapies, a simple pill is still incredibly attractive. It's cheaper to manufacture, much easier for patients to take, opens up a larger potential market.
Huge strategic advantages, especially for companies heavily invested in neuroscience, like Biogen, Takeda, Celgene, now BMS.
Absolutely. So when you see these billion-dollar headlines, it's not just about one or two drug candidates. It's pharma making strategic chess moves to secure access to a powerful long-term engine for drug discovery in this whole next wave of RNA medicine.
A very compelling rationale. But as always in biotech, the path isn't smooth. What were some of the major challenges Skyhawk had to overcome?
Oh, definitely not smooth. One of the first big battles was just scientific dogma.
The skepticism about drugging RNA with small molecules.
Exactly. For years, the prevailing wisdom was RNA is too flexible, too floppy. It doesn't have the nice, stable pockets like proteins do, where drugs can bind tightly and specifically. That was a major hurdle in convincing people.
And was that skepticism reflected in the lab work initially?
It seems so. They apparently had early struggles where compounds looked promising in simple cell lines, but then completely failed when tested in more complex, relevant cells like primary neurons. Finding molecules that worked robustly where it mattered was reportedly a really painstaking iterative process.
The classic translation challenge, from simple models to complex biology.
Right. Then, the next massive hurdle was taking that lead Huntington's drug, Sky0515, from the lab into human clinical trials.
The valley of death for many drug programs.
Indeed, and particularly challenging here. Huntington's is a rare disease, so patient recruitment is always competitive. Plus, regulators are, quite rightly, very demanding about safety data, especially for drugs designed to get into the brain. But Skyhawk did something quite clever here with their phase 1 trial design.
What was that?
Instead of just testing in healthy volunteers first, which is typical, they ran an innovative study that included both healthy people and patients with early-stage Huntington's disease right from the start.
Oh, interesting. Why do that?
It allowed them to gather safety data across different groups and critically get an early look at whether the drug was actually hitting its target, reducing that HTT mRNA in the people it's meant to help.
So getting biological proof of concept data much earlier.
Exactly. It was a calculated risk, but it paid off. It provided not just safety info, but that crucial early signal of target engagement and brain penetration. It was a massive validation point for the whole platform. Showed it wasn't just theory.
A strategic masterstroke clinically. Were there non-scientific challenges too, strategic ones?
Absolutely. There was reportedly this constant internal debate. On one hand, you need to keep building and refining the core platform, the SkyStar engine. That takes time and resources.
But on the other hand,
investors, understandably, often want to see tangible drug candidates moving forward quickly. Spin-outs, clinical milestones, things that create near-term value.
the classic platform versus pipeline dilemma.
Precisely. The founders apparently pushed hard for a more deliberate, slower approach focused on building a truly robust, reproducible discovery engine, even if it meant delaying some potential short-term wins.
And balancing partnerships too, I imagine.
Yeah, that's another tightrope. Those big pharma deals bring in vital capital and validation, which is great. But they also tie you to external timelines and priorities. You give up some autonomy.
So constantly weighing the benefits of licensing deals against the value of keeping programs wholly owned, maybe for a future IPO.
Exactly that kind of strategic balancing act. It shapes the company's trajectory significantly.
It sounds like a high-pressure environment. What was the culture like inside Skyhawk, navigating all this?
From what you gather, it sounds like a really interesting mix. Definitely mission-driven, focused on the patients, but also eclectic.
Reflecting the founders, perhaps.
Seems likely. You hear anecdotes about Bill Haney, the filmmaker's CEO, quoting poets or philosophers in board meetings, really encouraging creative thinking and risk-taking.
Okay, definitely not typical biotech CEO behavior.
Right. Then you have Sergei Pashkin bringing that rigorous scientist skepticism, apparently insisting on really tough, cross-functional data reviews, keeping everyone grounded in the science.
The necessary counterbalance.
And Kathleen McCarthy is often described as a sort of human connection hub, mentoring younger scientists, championing diversity within the team.
So different styles complementing each other.
It seems so. You even hear fun little stories, like chemists decorating the lab with origami cranes they folded from, like RNA sequence printouts, or the team staying late and watching a lunar eclipse together, reflecting on why they're doing this work. It paints a picture of passion, maybe some humor, and definitely resilience.
Sounds like a unique place. So, after all this journey, where does Skyhawk stand now? What's the next big chapter?
All eyes are really on the clinic right now, specifically on Sky0515 for Huntington's.
The lead program, where is it in development?
It's currently in a pivotal integrated phase 2 and phase 3 clinical trial. It's called Falcon HD. This is the big one.
Make or break time for the drug, potentially.
It really is. Positive data from Falcon HD would be absolutely landmark. It wouldn't just be huge for Huntington's patients, it would validate Skyhawk's entire splicing modulation approach in a late-stage setting. It could position them as the clear first mover for oral RNA targeting drugs in neurodegeneration.
A massive inflection point. Are they moving beyond neurology, too?
They are. Their next major program set to enter the clinic is Sky1214. This one is aimed at oncology.
Oncology? What kind of cancers?
Difficult to treat blood cancers initially, like multiple myeloma and certain types of non-Hodgkin's lymphoma. It's an oral drug candidate, IND ready.
And the preclinical data?
Reportedly very powerful, including observations of tumors shrinking to undetectable levels in some animal models. So this will be a really important test of the breadth of the SkyStar platform. Can it deliver in oncology as well as neurology?
Testing the versatility. Okay. And what about the company's own future? IPO aspirations?
That definitely seems to be on the horizon. With the pipeline maturing, the platform validated by all those partnerships, the company has been dropping hints about an initial public offering.
The next logical financial step.
Right. The goal would likely be to raise enough capital to take at least some of their own drugs all the way to market themselves, transitioning from primarily a discovery partner
to a fully integrated pharmaceutical company, controlling their own destiny.
That seems to be the ambition. Of course, they're not alone in this space anymore.
The competition is heating up. Other companies pursuing RNA targeting small molecules.
It is heating up. Several other players are making progress. Skyhawk's key differentiator, though, still seems to be that demonstrated ability to create oral molecules that effectively cross the blood-brain barrier.
That neurological edge.
Exactly. But ultimately, that advantage will need to translate into clear, undeniable benefits for patients in these late-stage trials to really secure their leading position in this whole exciting field.
A lot riding on the clinical execution now.
Definitely. So, in the end, it really feels like that filmmaker's instinct, Bill Haney's instinct, was right. The story of disease, it's not always permanently etched in our DNA stone. Sometimes the real problem is downstream. A typo in the transcript, an error in the edit.
And Skyhawk is essentially betting they've built the world's most sophisticated editing suite to find and potentially fix those errors. It certainly raises a fascinating question for the future of medicine. As we push into these areas, these targets once considered undruggable. Where do you, our listener, think the next unexpected blend of talent, vision, and technology will emerge to make the biggest difference in human health? What's the next frontier? Thank you for tuning into BioTales. If you enjoyed this deep dive into Skyhawk Therapeutics, Please subscribe, leave us a review, and send us your thoughts or requests for future episodes. And until next time, keep exploring the science that shapes our world.
The circuit hums with a heartbeat new. Tomorrow's edges in view. A world